I'd like Ms. Mamta Datla to say something about the company so that they know what kind of vaccines and how many variants are you making. Can you
Sure,
start
ma'am.
with Mamta Datla, please?
Of course. Thank you so much for the opportunity. So, first of all, our company was founded, like ma'am said, in 1948, and I won't get into detail, but we have a tremendous track record and history of vaccine manufacturing. But what I do want to focus on is the questions that we keep getting asked, and this is kind of a way to tell all my friends, that's the way I see it, what vaccines are and what are our choices regarding vaccines, what's the path forward in vaccine development. And I'm sorry if I'm, explaining this simplistically, but it's the best way I understand it. So I'm trying my best to explain it in that manner. So in natural immunity, what happens is in our lifetime, there's many pathogens, viruses and bacteria that invade our bodies. But our immune system is unique, it's miraculous, and we are designed to evolve in a fashion that our body doesn't recognize millions of these viruses and bacteria and sort of, don't even recognize it. But should one recognize it once an infection occurs, our immune system remembers this infection and creates an army of soldiers that says, remember this, we need to immediately take care of it. What a vaccine does is artificially creates that kind of attack system. It simulates the disease in a very micro-controlled manner. So it uses a piece of the same virus or bacteria or pathogen to stimulate the immune system in creating that memory. When I say artificially, I don't mean necessarily synthetically, but it uses the same bacteria or organism. So literally, that's what vaccines do. They simulate disease in a very controlled environment to get your body to do what it's naturally designed to do, which is create an immune response. The second thing, I get asked often, okay, so I have them, is, what's the normal pathway for discovery to launch of a vaccine? First of all, it takes enormous amounts of years of research and development, 7 to 10 years. And that is if you know what correlates of protection they are. That's if you know what works in giving protection against a particular pathogen. So you have proof of concept, you select an antigen and an adjuvant, and sorry to get technical here, but an adjuvant is kind of something that boosts your immune response. It's not a virus or bacteria, it's usually a chemical entity, but it gets your body to recognize the virus or bacteria to stimulate an immune response. Then what we have to do is animal toxicity studies. First, you have to make sure it's safe in the pertinent animal model, whether it's mice, whether it's guinea pigs, whether it's macaques or monkeys. And then we conduct one clinical study where we test the safety in adults. Remember, vaccines are designed to be given to infants. So the highest safety standards are practiced in terms of clinical development and safety of a product, because you're not giving a product to somebody in late stages of cancer where you can take risks. You're giving this to healthy infants that everybody is looking forward to protecting and having productive lives. So first we do what is phase 1 clinical studies, which is you study the dose in adults to study whether it's safe. Once it's safe and tolerable, then you go into phase 2 and phase 3 studies. Now, typically phase 2 studies are where you select the dose. You need to know if 5 micrograms of the virus is enough. Do you need 10? Do you need 20 in combination with adjuvants? In adults it works differently. Children it works differently. So this is the phase where you decide which dose is good. And the third phase of clinical trials, there's 2 components. One, where you don't know it's no pathogen. Then you do what people are doing now, which is called efficacy studies. Basically, you give huge numbers of people the vaccine, and then you have a control group that you don't give the vaccine, and you have to statistically that the disease occurs more in the group that did not receive the vaccine. Then there's a second group where previously known vaccines, you just have to do a comparability study. But this is the extensive process we go through before we do market authorization and launch. And while this is going on clinical development, we have to scale up the process, make sure the process is consistent, make sure we can make it safely, make sure we can affordably, and so on and so forth. Now, I'll just briefly talk about the types of vaccines that are currently being discussed. One is mRNA or DNA-based technologies, and I'll combine them because they're sort of similar. It's basically like giving your body code. And for all the IT people out there, this is how I understand it, even though I'm not an IT person. It's like writing code for your body. And it's instructing your RNA cells or your DNA cells to produce a certain type of protein that will inhibit this pathogen from entering the body. And the classic examples of mRNA are Moderna and BioNTech Pfizer. There's a DNA-based vaccine in phase 3 by 𝗕𝗏 in India. Then there's a third class of vaccines, which are inactivated viruses, where you take the whole virus and you basically weaken it or kill it a little bit. A little bit like Schrodinger's cat, it's not completely dead or alive, but you weaken it by chemical or heat, and then you introduce that into a person, and your body recognizes it, but is not overwhelmed by it because it's weakened and produces immune responses. And that's Sinovac, it's Bharat Biotech's Covaxin is an example of that. The next one is the non-replicating virus vector. And this is really where J&J, AstraZeneca are classic examples of this, where you take the whole spike protein, but you need something to carry that spike protein into your body. And your body doesn't recognize that by itself. So you need a carrier. And the carrier in these cases is something called an adenovirus vector, which is already present in our bodies. It's a cause for common colds and so on. So we use that virus, which is harmless in theory. And then on top of that, we put the spike protein of the coronavirus, and this harmless virus actually carries it and presents it to our immune system. So like I said, AstraZeneca, J&J, and through our collaboration, as I suppose, Sputnik are all examples of this. Then there's a class called the protein subunit vaccines. And this is an established category where the carrier is not a different virus. So we just take a subunit protein of the vaccine and we express it and give you just the protein. So there's no third DNA being introduced into your system. And hepatitis B vaccines are a classic example of the success of this technology. Our partnership with Baylor is a good example of this technology. Novavax, Clover, Medicago, Medigen are some of the companies working on this technology. And the last class of vaccines is what we call attenuated live virus-based. And this is basically you take the whole virus and you passage it, or you let it grow multiple times, where you keep picking the weakest of the family, so to speak, the weakest of the virus, and that's the one you end up growing. We have several live viruses, vaccines as examples. Measles is such, polio can be attributed in some cases as such, rubella and mumps, but this has not been chosen as a way to go for COVID because it's a very high bar to prove safety.